I don't know if it's working or not. This one, it's working? It's good? Okay, good. It's good or not? Okay, good. So it's my pleasure to give a lecture here at IUPUI. So right now, I mainly work on metagenomics, just to apply next generation sequencing to study the genomics of a whole community of microbial organisms. So it's genomics, but the genomics is applied to not an individual organism, but a whole collection of organisms that you can collect from anywhere, essentially anywhere, from human bodies, from ocean, from anywhere you can think of. So I don't really know, I don't do sequencing, so my job is to give me a sequence, and I will start from there. So I will not tell you how many nanogram DNA is required for sequencing for metagenomics, because I really don't know for sure about that. So I will not talk about the details of the experiments for metagenomics, but I will focus on the computational side. Once we have a huge number of DNA sequences, what we can learn from those sequences about two aspects: the taxonomic composition and also the functional content for a whole community of microbial organisms. So it would be impossible to talk everything about metagenomics. I'm also still learning. I think it's really a field that's really getting broader. But I will just try to give, within this 1 hour or 1 hour and 15 minutes, I will talk a little bit about the background. And I will mention very briefly about these three very exciting metagenomics projects, the huge ones, the MetaHit, HMP, and also the EMP. So I will mention about these three because I think it's good to know at least those names. And I'll focus on computational methods. What are the methods that are required to analyze those metagenomic sequences? So, but whenever we talk about methods, you need to know what you care about a whole community of microbial organisms. So if you only care about what are those organisms, so it's E. coli, it's streptococcus. So you just want to know the taxonomic information from the metagenomic data set. Probably you will do different experiments. You will only get 16S RNA gene fragments from those samples, and then you will do some analysis of those 16S RNA gene fragments. But if you really care about the functional content of a metagenomic data set, so you need to do shotgun sequencing. So if you get a sample from a human body, you want to do shotgun sequencing. You want to get reads from all different kinds of genes from that whole collection of genomes. And from there, you want to guess what might be the functions of those microbial organisms they encode. So what they do. So there are two questions: who are they? And the second question is what do they do? So if you just care about who are they, then you can just look at 16S RNA genes. But if you really care about what do they do, you need to do shotgun. So
actually, metagenomics is RNA part.
No, both. So it can be 16S. Depends on your purpose. So the previous ones, mainly because the next generation sequence was not so cheap yet. So you may just focus on 16S, because then you can go very deep if you only get rates from 16S RNA genes. But now the throughput is so high that you can afford to get rates for all the different genes in a sample. So you need to do shotgun to get rates from all the different genes from a sample. So if you have questions, just stop me. I think it's really not just some seminar or whatever. So yes.
But if you want to know the functional,
all the DNA sequences.
All the DNA.
Yes, all the.
Sequence the DNA.
Yes. So you can try to guess what do they do, just like human genome. So once you have human genome, you can look at those, for example, the genes encoded by the human genome, and from there you might guess what are the functions that human genome encodes. So it's not really. There are meta-transcriptomics projects as well. So for those projects, they care more about if a gene is really expressed in that environment. So here it's just possibility. So you can see those DNA pieces there. And this is an enzyme encodes for a kinase or encodes for ribosomal RNA. So you know the potential of those, you know the functional potential for a whole community, but you really don't know if the gene is expressed or not. So that's some people argue about. So like one of the metagenomics topic is to get DNA from soil samples. So essentially, you can think everything can eventually get into the soil, the DNA. They don't necessarily live there. They are not functional there, but it's just because they're dead anyway, but they're degraded into the soil. So you will have traces or evidence of DNA of those species in soil sample. But some people argue that they're not really not functional in the soil, so you should not consider them as organisms that you found in soil. So that's a really good question. You just get DNA pieces, you know its potential. There's this community has this potential, but you really don't know for sure if this gene is functional or not there. So, but worst case, you need to do a meta-proteomics. So even though you do meta-transcriptomics, you know this gene is expressed, but you still don't know if there's some proteins produced from those transcripts. So they are meta-transcriptomics projects and also meta-proteomics projects, but they're not as popular or common as metagenomics because of the next generation sequencing. It's so cheap. It's so convenient. So just stop me anywhere if you are confused or you want to know more. And also I will show some of the resources that might be useful for you if you are really interested in metagenomics or if you want to do some research in metagenomics. And I want to tell a few stories about what you can do, what you can gain by looking at those large collection of DNA sequences from a mixture of species, a mixture of organisms. But just a few stories. And also, if I have time, I will go over a couple of computational tools that we have developed for metagenomics analysis. So maybe just to mention a name or a few names, but if I have time, I can go over a couple of them. So, metagenomics, we usually do analysis of a whole collection of organisms, microbial organisms. So you don't do metagenomics for human beings. You can just do individual human beings. The microbial organisms, you sometimes you cannot get, you cannot isolate those ones. So you only have a whole community. Like there are tons of microbial organisms in your gut. There are tons of microbial organisms on your skin. And the most important thing is that those microorganisms, you cannot culture them, you cannot grow them in the lab. So ideally, you probably want to really go through each of them one at a time, so that you can do the traditional genome sequencing. You can sequence each of those species, and then you can analyze the whole community. But here is that you don't try to isolate those species. You just try to collect DNA from a mixture of the species. And also, that's one thing, because that makes it possible to study the DNA material from those microbial organisms. That's because we can do that now. That makes it possible, but also it makes sense because those microbial organisms, they do really work together as a whole community. So if you analyze those individually, you don't really get any insights into the interactions between those microbial organisms. So one side is that it makes possible to do analysis of the DNA genomic DNA sequence from those bacteria, and also the other side is that it's possible that you can analyze them all together as a whole.
What is L9? With bottom?
Light year.
Light year.
So it just give you some idea how many of those cells are there, they live with you. So it's just the other huge human genome. The other human genome is not really your own genome, but it's really, you carry a lot of genes just because of those microorganisms that live with you or inside of you.
10 times more.
You have 10 times more cells, although you don't really see them because the cells are smaller. So those bacteria cells are smaller than your own cells. So even though you have tons of those tiny organisms living on your skin, you don't really see them using your eyes. But there's so many of those that you cannot ignore them. So, and also because why we care about those microbial organisms, sometimes because we're just curious. We want to know what do they do. But I think people get to know about those microbial organisms because they cause troubles. They're pathogens, they cause disease, so they're bad ones. But now it's really, scientists are realizing that we do need to have those bacteria living with us because they're commercial, they're beneficial to our health. So they could cause troubles, they could cause disease, but they also are beneficial, many of them. So just list some examples that sometimes those bacterial organisms or virus, they can really cause a lot of disease, they cause troubles, but they're really good ones. So and that's really one of the reasons NIH initiated this human microbiome project. So that we can get some idea about what species living with human beings and what do they do, what's the impact of those microbial organisms on the human health and the human disease. So that 10 times more is just cells. But if you count the genes, it has more because different organisms, they carry different genes. Essentially, all the human cells, they have the same set of genes. Although they may have different expression or whatever, epigenomics. So, but essentially, all the cells should have the same set of genes. But those microbial organisms, they don't have the same. They have different genes. So if you just count number, it's 10 times more. But if you count by genes, it has a lot more.
If you go back to the table, I think it's 3 times 10 power 6. Is that
This one? The genes. You need to count the genes.
Yeah, it's 300,000.
Right. That's only for gut.
Only human gut.
Human gut. So if you include other sites, body sites, it will be more.
So human gene is about 2 times 10 power 4. So it's 100 times more.
Yeah, just for gut. Just for gut. Mm-hmm. So essentially, instead of doing one bacteria at a time, so like here, what scientists did for this traditional genome sequencing project, you get you isolate the bacteria, you grow them in the lab, then you can do sequencing, so that you will hopefully for like E. coli, you get a circular genome for E. coli. But the bacteria, they have to be able to grow in the lab. But But it's not always true, as I said. Some say like they're more, the majority of the microbial organisms, they cannot grow in the lab. Maybe because we really haven't figured out their requirement. We don't really know how to grow them yet. But the percentage is very high, no matter what you do. So a lot of the bacteria, you cannot really grow them in the lab. So that's the reason many scientists just go out to nature to get the sample, and then from those samples you can extract DNA. From those samples, just like could be a drop of water or fecal samples. If it's human being, you're interested in the organism living in the gut, or just swab the skin and you can get some samples. From those samples, some labs can extract DNA from those samples. And those DNA samples will be sent to a sequencing center so that you will get a lot of DNA sequences from a mixture of microbial organisms. So that's the whole point. Instead of getting a rates from a single genome, you get essentially DNA rates from a collection of different genomes. So that's I think those are the pictures from Venter's project. So the Global Ocean project. So they try to survey the microbial diversity, like in different ocean locations. So you do expect they will be different. So they are trying to do a global survey of the ocean microbiome diversity. And I think that's is that? Oh yeah, one of those are really Venter's private boat. So I think when was that? I talked to one of the guys working with him, and he said the boat is very nice. So
the boat is nice.
A very, very nice boat with everything. So with this technology, or because of the again the advance of next generation sequencing, so it's possible that we are seeing the genetic material of a variety of microbial organisms. So before we cannot isolate those, so we don't know, or we cannot get those genome sequenced, but now at least we can get them in some way. So I think for now, I think the two major techniques that are used for metagenomics, to my knowledge, is one is the 454, and the other one is Illumina. So 454 is mostly used for 16S based only. So if you care about the composition, what are the bacteria in a community, you can just go with 16S. You only care about 16S, you get 16S. So for those, if you do 16S, you do want to get a little longer rates. So 454 gives you longer rates. And also you don't need to have.
How do you only focus on 16S? You have some characteristics?
Yeah, I will show that, but mainly you can use primers that target against 16S only genes, so that you only amplify genes from 16S, and then you will do sequencing. But Illumina is just for shotgun metagenomics. I think Illumina is the main one. Even though, like for human HMP project, the original proposal was to use 454 for the shotgun as well. But then Illumina was, I think because the throughput of Illumina is a lot higher than 454, so that I think it's just halfway through using the 454, then HMP, the whole project, switched to using Illumina sequencing for shotgun metagenomics. But metagenomics, that's not the only definition, but it's just a study of the entire genetic material of a whole community of usually bacterial organisms but those microbial organisms, those tiny organisms, and they could be from anywhere. Essentially from anywhere, as long as you can get a sample, you can get DNA extracted, you can do metagenomics. So a few more definitions, like a microbiota is just the whole collection of the microbial organisms of a particular site or region. Like human gut microbiota is just all the microbial organisms you can find or living in the human gut. And the metagenome is just the gene content of all those organisms living in one site. And also, as I mentioned, the metagenomics just to study the genomic material for a whole community, of microbial organisms. So mostly people care about, or doing metagenomics, we do prokaryotic organisms. But there are some groups, they also care about those small eukaryotes. So that's a little bit, a very short background on metagenomics. And as I said, I will mention these three huge metagenomics projects. The Earth Microbiome Project is the newest one. I think it was initially this year or DOE. But actually BGI is contributing like 75% of the funding. So I guess they will just do the sequencing. And Argonne National Lab is also heavily involved. So I think probably they will do the analysis of the sequence. So they will do like 200,000 samples, microbial organisms from soil, air, sea, and freshwater systems. So they don't do human or other animals related. But I think most of them will really from soil. And I think they already have collected a lot of samples. But they will do both 16S and also shotgun. Even though Illumina is great, but you can really not, probably it will be still very expensive if you do shotgun for all of them. So, but you can afford to do a 16S survey for all the samples you can get, and then for a selection of the samples you can do shotgun.
So when you talk about 16S, you mean if this is a group of soil, if I have, let's say, 10,000 species in there, they have differences in the 16S.
Yes.
Right?
Yes.
But you mentioned that you want to do the amplification design primers. How does that can work?
Yes. So 16S RNA genes, they're the most conserved gene family. So they're very conserved. That makes it possible so that you can design some primers so that you can get 16S from all different species. Not really all, but close to all. I will show the structure of, I don't know if I will show the structure, but 16S, they do have very conserved regions and hypervariable regions. Yes, so you will design the primers against those conserved regions so that you will have primers against the conserved regions, then you will be able to amplify both conserved and variable regions. Yes? I think it's degenerated primers, a collection of primers. But there's some bias. I think people are studying the bias. As I said, probably they are not equally good for all different species. I mean, some primers are better for this group of species. Another set of primers might be better for a different group of species. But the general idea is that we can use degenerated primers that can target against those conserved regions from 16S RNA genes, so that hopefully you can you will be able to amplify rates, gene fragments from all different 16S RNA genes from all different kinds of microbial organisms. So that's what we hope. But you do really expect to have there will be some bias in those data sets you get. So as I said, the EMP includes BGI because they will do the sequencing. Argonne National Lab, they will do, I think probably they will do the analysis of those data sets. So this is one of the newest, and probably will be the biggest one, because they target so many samples, and they will do both 16S and shotgun. I think they even worry about how they are going to release those data, because there will be a lot of data. And some people say they should release the data, and then others say if they release data, there will be very few labs or groups can really handle such a large collection of DNA sequences. So I think it's still very new, because there are not really much results or discoveries from this project yet, but it's just something that you may want to know. So MetaHit is probably one of the oldest big project, metagenomics project. So it's European-based. So they really want to, they mainly do metagenomics for human gut samples. So they don't do other sites, just human gut, and they want to get some idea about what are the bacteria living in the gut, and what's the relationship between those bacteria and a few human disease, like the or the common phenotype, like the obesity. So if there's some relationship between those microbial organisms and obesity, or a few other, like a disease. So very few of those diseases. And that's the paper published last year in Nature. So that, I think that was the first publication on that, on the large-scale metagenomics project of human gut. And you can expect you will get a lot of genes, including new genes, from that metagenomic, from those metagenomic data sets. And also, there's another new paper coming out from the same group of scientists. So they can classify humans based on the composition of their gut microbial organisms into 3 classes, 3 types, they call enterotypes. Although it's really debatable, but it's just something interesting.
What's the sample size?
This one, they use several hundred, I think, Sanger rates. It's very interesting. They use Sanger rates for the second paper, the enterotype. And they also check with rates. They also use the rates from Illumina sequencing. Although you can still see like 3 types or 3 clusters of samples. But it's not really that obvious as compared to the analysis using Sanger rates. But I think right now you don't really see that many metagenomic data sets of Sanger rates, because the throughput is really not high enough.
We got get a patient and get a gut and some kind of parts and do the. But is there any relationship between the microbiology species in the gut with the patient itself? So they really, the genes or the phenotypes, do they really communicate?
There's some correlation. So that's one of the goals for the NIH Human Microbiome Project. Try to figure out what's the impact of those microbial organisms on human disease. So there are not so many really discoveries yet coming out of those ones. But what we do find is that there's huge variation among individuals. And like you, me, or everyone here, we really have very diverse collection of microbial organisms. But you can still tell if this DNA sample is from gut or from skin, because they're very body site specific.
And once you got a mixture of all the species in the gut, for example, so is that the answer is you have this or not, or is that complicated answer? So if you say I have this species for this much and that species for that much, is that?
Yes. So if you already have those genomes sequenced, you can definitely do that. You can either you can do quantification, but it's really complicated. I will mention a few those challenges there. How to make sense of a mixture of DNA sequence from different organisms. So MetaHit is just for human gut, and the human microbiome project supported by NIH. So I think it's into fourth year. And the aim of this project is to characterize the microbial organisms in very different body sites, various of body sites, of course including gut, because the gut sample is very easy to get. It's not really gut, it's a still sample. But the HMP also looks at oral sites, multiple oral subsites. So it's not really, the whole mouth is not just one thing. Teeth, tongue, and they're still different. The multiple oral sites like skin and other sites. So I think, but the most samples are really from, you really have, I'm involved in HMP projects, so I do some analysis of those data sets. So you can see a lot of samples are really from oral sites because they're easy. They're just easier to get. And also a lot of gut samples. NIH.
Yeah, which one?
I think the human genome, that one. It's a roadmap project. So I don't know maybe in general one, yeah. And also they try to analyze the role of those microbial organisms on human disease and health. As I said, they are really important. We cannot germ-free because then you will be in trouble. So many of those are really good ones. We do need to have those ones. But some of them, they do cause troubles because of disease. So there are some projects, they just focus on a particular disease. And there's some indication that some of the microbial organisms may have something to do with those diseases, then you can do control normal comparison. So that's just a few sentences showing why we need to have those microbial organisms, because our immune system, one example is that our immune system needs to be trained by those microbial organisms. So these are, as I said, there are mainly two questions for now. What are the species? What do they do? So it really depends on your purpose. Some projects will just limit to 16S RNA gene-based survey, but now I think most projects will just do both because they really gives you different insights. For shotgun, for the rare species, probably you will never be able to get anything from those ones. But if you only do 16S, because you enrich the rates, so that it's possible that you may still get 16S from those very rare species of very low abundance. And there are different challenges, analysis challenges. So once you get a lot of those 16S or a lot of those shotgun rates, so the first problem is that there's so many data. I have servers, but each time it's always like a reach to 100% full. So it's really, you get so many data, and it's really even difficult to read in those huge files, not to mention to analyze those rates. So I will show some of those computational problems and also the challenges. As I said, but there will be different types of algorithms for computational approach for these two problems. So if you want to figure out who is there, or if you want to figure out what do they do, you will work with different data. Probably you need to have different algorithms for these two different purposes. So 16S. So this is the 16S gene. So you can see from here. So it's about 1,500 base pair long, those genes. And if you look at the conservation patterns of those genes among different species, they will have many very conserved ones, and also like those very hypervariable regions called hypervariable regions. So ideally, you really want to get sequences from those variable regions so that you will be able to differentiate what species are there. So, but you need to amplify those out. You need to amplify those regions so that you can design primers against those conserved regions so that you will be able to pull out those variable regions. And one of the difficulties is that you don't really get the full length of 16S because there's no second generation sequencing. Techniques can give you like a 1,500 base pair long rates. So as I said, 454 is the one is the most commonly used technique for sequencing 16S RNA gene fragments. So that's usually people will get design primers like this one, so that you will get fragments that cover region, hyperregion 1 and 2 or 1, 2, 3. So that you can get those amplicons and send them to the sequencing centers. So that those sequence centers will be able to give you rates of about like 400 base pair. So hopefully we'll cover some of those hypervariable regions, but definitely not the entire length. So there are.
I'm just curious. There's so many different microbiology species. Are you assuming that all the 16S has this structure?
Yes. There, as I said, 16S is one of the most conserved genes.
As long as these bacteria should have
They should be similar in some sense. They should have very complicated structures with stems, with loops, and also those conserved regions. You will even be able to come up with primers so that they will bind to all those regions. So you may miss some of those, the very strange ones, but hopefully you will get most of them. And you can put tags into those amplicons, so that you can just run one sequencing, but you can do like thousands of samples, just one at the same time, because you can separate them into different samples based on the barcodes. So that's really why it's called barcoded pyrosequencing. So otherwise, you need to do one sample at a time. The throughput is so high that you can really do multiple samples at a time, so that you will.
Ideally, how many sequencing rates? What's the coverage for this?
So I think it really depends on the complexity of the sample. So some environments, they have very high complexity. You have thousands of species, and you can guess what do you need to have. So some people will go very shallow sequencing. So that was for those you will only get 16S from those very abundant species. But if you really want to get the rare ones, you need to go very deep. So Rob Knight, he really likes to do shallow sequencing. So he will get like several thousand reads, just several thousand reads for a sample. And sometimes that's good enough for for some purpose. So if you want to distinguish like gut versus skin, probably you just need to have the first 1,000 reads, because the abundant species will be different.
But they have to prepare libraries for each sample. It's not very most.
Yes, they need to, but I said they can put different barcodes,
Right.
so that at least they can do the sequencing at the same time. But they still need to do individual sample. Right. So why it's so difficult? I mean, once we get those 16S RNA gene fragments, we try to figure out what are the species there by just looking at those fragments. As I said, it's not a full length. You only cover a part of the gene, maybe one third or even worse than that. Usually you just get several hundred base pairs. And the entire length, as I said, is 1,500, about that. So also you have a lot of like noise. 454 probably has higher sequence errors as compared to Illumina. I don't know, all equivalent, about the same. But they do cause a lot of problems. 1% sequence error can cause a lot of problems because 16S genes, as I said, they're so conserved. So 1% difference, you may consider they're from different species. Yeah, and they have different types of errors. So the problem is try to get, once you have all those rates, and also they contain errors, you try to figure out what are the original species that those rates are amplified from. So there are two types of, generally there are two types of methods that we can use to figure out what are species there. So the first one is taxonomic assignment-based method. So essentially, you just map those rates back to the known 16S RNA genes. So you can do alignment or whatever method you can think of.
So then you have to have a database of all of those.
Right. There are several databases out there. One is the Green Genes. So Green Genes database. So you can just search your database, your data set of 16S gene fragments against Green Genes. Because there are so many rates, you cannot just do Smith-Waterman alignment. So that's reason the Green Genes team, they come up with this Rdp classifier, which is really a naive Bayes-based method just by looking at the frequency of chimers. So you don't do alignment, real alignment. You just look at the chimer frequency so that you can hopefully assign one rate into one of the clades. Actually, they can only do up to the genus level. So I can assign this 16S gene fragment is probably from that genus. Usually you don't go below that because the confidence is not enough, the support is enough. You only have fragment, and also because Rdp Classifier is, as I said, based on the chimer frequency, they don't really do alignment. But if you run BLAST or you use other similarity search tool, you can probably just, if there is gene, if this fragment is really from a gene, the 16S RNA gene of the known species, probably you will get almost perfect match. So, but for this one, you really limit yourself to the known species. And as I said, one of the goals is really to figure out what's the diversity of microbial communities in a sample. So you really want to go beyond those known species. And so that's the reason some people will look at, will try to cluster those 16S RNA genes. And from the clustering result, they try to figure out how many possible species are there in a sample.
How big let's say 16S ribosome RNA records are? I mean, not for one species, but because you have to do alignment against all of those. Right?
Yeah, I think Green Gene is huge. because 16S, you don't really need to have the whole genome sequence to get that one. So there are many people do PCR and to get those 16S.
How big not the query part, but
the database is huge, but I don't really remember how many sequences are there.
Is it comparable to the human side, or?
It's not comparable to the humans.
Not as very
I don't really know. I think green genes is quite big. I don't remember the exact size. But you can still. But if you really just want to run blast, that will still take quite a while if you have a huge query data set. So the second approach is just try to cluster those 16S RNA gene fragments into clusters. So you will cluster similar fragments into one cluster. So hopefully, all the fragments you cluster into one cluster, they present one species, for example. So they called operational taxonomic unit. So it's not exactly a species, but it's close. So like if you use like 97% identity as a cutoff, so that you want to make sure everything you group in one cluster, they share at least 97% identity. And you will get they are probably from the same species. That's the species level of resolution. So 97%, again, that may change for individual cases, but that's the criteria people use for defining species. So a genus, you can use higher cutoff, but anyway, you can just use a cutoff of 97. What will be the how many OTUs you will get if you use 97% cutoff, similarity cutoff as a cutoff. So, but essentially, it's a clustering problem. You can the mother use like average linkage clustering, but the problem is that it's really for all those clustering algorithms, it's like n squared, the complexity n squared. So if you have so many reads, you cannot even do clustering, those average linkage clustering. So for the HMP project, there are so many reads. Probably only one or two groups can do that using supercomputer, so that you can run all the pairwise, essentially all the pairwise alignment, and to load in the pairwise distance matrix, so that you can do clustering. So that's really I cannot do that because I don't really have a powerful enough computer for that one.
So powerful meaning the memory, or?
Memory and also computational time as well. I think
Mason
but
is not enough to have half the terabytes.
I don't think Mason is enough. Because you have this pairwise distance matrix, essentially you need to load in. Even you can use other tricks, you can use sparse matrix, but it's still a lot of pairwise distance. So that's the reason some people just don't do clustering anymore. They will use those heuristic methods like CD hit. So you will just random pick up one and use that as a representative sequence and group others to that one as long as they are similar. So it's not really a rigorous clustering algorithm, but it works a lot faster. But if you use those heuristic methods, you will get you will overestimate. Therefore, you will overestimate the OTUs. But if there's nothing else you can do, probably you have to live with those ones. So ChIP. ChIP is one of the newest one in this field. So if you really need to analyze 16S, a lot of 16S RNA rates, probably you can look into this ChIP. It's from Rob Knight's group. He's one of the biggest guys in doing 16S-based survey. But actually, it's a wrapper. It really wraps everything. So it does provide analysis for taxonomic assignments and also OTU-based analysis. And also, it provides a very nice visualization of the results. And they also provide a virtual box because they include so many third-party softwares. So if you want to install, it will be even difficult to install that package on your own server. So so that's one of the reasons they just provide you with one virtual box, so that hopefully you don't get into trouble when you try to install their system to your local system. So but it really has everything, essentially.
So they created kind of their own cloud environment that you can
No, they don't provide service. They just provide this virtual box so that you can.
And you can push that to your computer or to.
Yeah,
cloud,
yeah,
right?
yeah, yeah. So, but they don't do service. And they use very different metrics for measuring, for example, the distance between two communities. Yes.
So for work with December 4.4 and the Unifrac what happens to solid? The Metagenomics community is solid
I don't see any solid data for Metagenomics yet. I said mostly are 454 or Illumina. Sanger, very few, the older ones. And this package allows you to analyze the diversity or like evenness, whatever, of microbial communities. So one of the metric I just want to mention very briefly is the Unifrac Distance. So you may want to sometimes, especially like computer scientists or informatics students, you want to really come up with a number to measure how similar these two communities are in terms of taxonomic. So you want to come up with a number to measure the taxonomic similarity between two environments or two communities of microbial organisms. So this is the metric proposed by Rob Knight. So essentially, you will put all the reads from Rob Knight.
Is that the same thing as the basketball coach?
Maybe, yes, Bob. But he's not the one. So for this Unifrac metric, to calculate this one, you need to put everything into one tree, so that you can look at the branches, the unique branches that unique to one of the communities. So like the one on the left, So essentially, these two communities, the red one and the blue one, they don't share anything. It looks like they don't share anything. So this environment will, the blue environment will have this branch, and the red has the other branch.
So one block is one, for example, the species in my gut, and one block is one square is the one person.
So here you can think one block, one is just one read, or many reads. So, but all those reads can be assigned to this branch. So this is a tree. That's a phylogenetic tree that reflects the evolutionary history of those relationship of all the reads. So which means the blue environment has very different species as compared to the red one. So all the branches are unique. So Unifrac is just the percentage of those unique branches among the entire length of the tree. So the left one, the distance will be 1. So which means they're totally different. And the right one is about half of that. So they have some unique ones, but many of them they share branch.
Yeah, so each square is what?
You can consider that's a species, but and then you can think that I will, for example, I will assign like 100 reads to that square, and there will be 1 or 2, so it can be weighted or not weighted. So if you don't consider weights at this moment, just ignore the abundance. We just care about if this species is there or not. So if it's blue, so which means the blue environment has this species. And the red ones are the same. So this is, you can just consider it's a phylogenetic tree. So, but this one is a lot, they're a lot similar. At least this branch is the same. So they only differ slightly here. So So for this one, you consider this, the evolutionary history of all the species, as compared just to count how many unique species. So it's very nice metric for measuring the taxonomic similarity of two communities. And use this one. So if you have like 10 communities, like 10 samples from 10 individuals, human beings, and you can try to cluster them. Or if you have 10 from human gut and 10 from soil, and you can calculate the pairwise Unifrac distance, then you can do clustering. Probably you will get 2 clusters, one for human gut, one for soil. Usually, if you go with that extreme case, you will get very nice clustering results. Yes, very nice separation. But this is metric so commonly used. Just I watch, and it sounds very straightforward. So I just want to mention this metric briefly. So that's for 16S RNA gene fragments analysis. So but now people care more about the functional content of a whole community. So we need to do shotgun, and then we need to look at all the different gene fragments from the community. So, but once you have the a mixture of DNA sequences from many different genomes, and what you can guess from those ones. But definitely, just like what you do with a single genome project, sequencing project, you want to assemble them. Ideally, you want to assemble them into individual genomes. Like if I have 10 genomes, 10 species in one sample, ideally, I want to get 10 genomes. But usually, you cannot do that. It's impossible. So, but sometimes you may get like a very good assemblies for the abundant ones. So it's possible. So you may get a few genomes, but most of the time you will just get short reads, pieces of DNAs from many genomes. So that's assembly. So there's no perfect sampler for metagenomes yet. And the super de novo is used in MetaHit project and also in the HMP project.
So why the assembly of metagenome is different, so much different?
It's not so that different. Ideally, you still try to use the overlaps between the rates. But if you work with a whole community, you can think like there are many species, they are similar to each other. So they're just like pseudo repeats.
You're saying that so de novo is the most often used one. Is that because most of these data was produced by BGI?
Maybe it's just because it was first used in MetaHit. Because BGI was involved in the MetaHit as well. So super de novo was used that one. And also it's the Boolean graph-based method for assembly. So most of the assemblers are based on the Boolean graph approach. And this one works. It's not a lot better than others, but it works fine.
Because we had the RNA sequencing lecture last time, and there were de novo assembly on the RNA sequencing, and the comparison was so de novo is by far the worst assembler.
But for transcriptomics, that's a different story. But for the metagenomics, it's essentially try to assemble genomes. So it should not be that different from, like, for example, you try to assemble an E. coli genome. But the complexity is higher because, as I said, there are many similar species in the same sample. So they just like repeats. So they really confuse assemblers. And also because of the abundance difference. So you have some genomes are very abundant and some species are very low, of very low abundance. So it's very hard to distinguish those ones if those are just because of repeats or they're from high abundant species. So like if you just assemble human genome, you know if like this region is really highly sampled, probably it's a repeat. But in metagenomics, you don't really know if it's. Yeah, for that sense, that something could be similar.
Yeah, I'm not questioning this. I'm just curious. Why so always one?
At least for those big projects.
Probably because DJI was involved in the reference.
Yeah, I think also the HMP analysis team, I was also involved slightly. So we do try to different assemblers. And there's really no champion. It's just this works, and also it does not use that much memory. I think Velvet used a lot of memory. So Mihai Pop, he's the one that leads the assembly effort for the HMP, and he said a metagenomics assembly is impossible. That's his title. He gave a presentation on the